This is a microbiome test that Dr. Mohr, who's on the call with me today, has been developing for quite some time now. And a lot of work, a lot of elbow grease has gone into this to make a really, really good microbiome test. What can you expect from today? You see this nicely designed report with a microbiome panel here on the right side. But what we're going to be showing you today is going to have utility regardless of if you get into our panel or not, that we are doing an early access launch for today. I think this is going to be helpful information for just understanding the landscape of the microbiome and how it correlates to human health. So what you can expect to get out of this webinar by the end is a deeper understanding of the significance of the microbiome on human health, how to utilize AI in microbiome analysis, understanding the function of the gut, not just taxonomy. If you saw that one email, I kind of went through like, hey, it's not so much just about this specific species or this specific biomarker. It's really about the function, right? Because we don't care so much about the species that are there. We really care about how they function and how that correlates to overall health. So we're going to be getting into like, how do we come up with that? How do we understand the function of the gut from the species? And then getting the most information possible from a single test. But what I've seen Dr. Mohr do over the last year working on this report and this analysis has just been, it's really blown me away a few times in regards to what he's produced. And so I'm really excited, not only for the depth of information that he provides within a report, because that's one thing is depth of understanding, but then understanding how to translate that and communicate that to a patient and understand their health and their landscape is an entirely different thing, because I can give you a bunch of biomarkers and all their values and that kind of thing, but then you have to be able to translate that and understand how does this correlate to human health. And so we're going to be getting into some of that today. So who are we? As a company, we are Theriome. Today, it's going to be myself. I'm just doing this introduction, and then I'm going to be handing it over to Dr. Alex Mohr. He's an expert in microbiology. So he has done quite a bit of research on the microbiome, how it correlates to human health. And he actually gets some pretty big research contracts from I believe the NIH, and I'll let you speak to that, Alex, but just some really impressive research that he's put out in this space. And not only that, because as you know, researchers can be kind of off in this data thing, but how does that correlate to clinical relevance? How do we make action on that information? And so he has a really good blend of those things. But as far as me, my name is Micah Lowe. I am a partner and the CEO of Theriome. I got into this space because my brother had brain cancer, and my goal at that time was to find things that would be helpful for him. So I have this kind of long backstory. I'm going to make it really short. Essentially, I was looking for things to help my brother get better and see what I could do. Found some things that were massively impactful for him. He ended up living 16 years longer than a lot of physicians anticipated, but that really, from a pretty young age, set the tone for what my trajectory has been thus far. And I've kind of summarized it in that my goal in life is to reduce suffering in the world through precise, non-toxic medicine. And so I see Theriome as an excellent opportunity to do that, because what I noticed in some of the other businesses that I have is that, or I go to a conference even, they'll have a modality or they'll have a thing, and then they're kind of a hammer where everything is a nail. Like, I do this one thing and it works for all these different things, right? And so what patients end up struggling with is, where does this fit into the context of my health and my narrative? Like, there's so much information overload, and everything is like the best thing since sliced bread, right? And so then how do you begin to parse that out and understand what is actually going to be the most impactful for you? And that's really the mission of Theriome, is how can we understand the biology of the patient on an individual level and then give them recommendations and insights on exactly what to do. I think in the future, this is going to include medications, even things like chemotherapy. Does chemotherapy even work in your condition, in your state? Those are things that we'll be able to uncover. And so we'll be able to give insight into what people should do and what people should not do, which is equally as important. So our mission at Theriome is to deliver unparalleled health insights through advanced multi-omic analysis and empower providers with personalized protocols for their patients, offering a better mechanism for standard of medicine than currently provided. There's a little bit to unpack there. Alex will be going through some of that. If you're not familiar with multi-omic, that's okay. You don't need to to have a deep understanding of that. These pictures are just to explain, hey, we're not a ragtag group. We do actually have a lab. We do actually do analysis, a very state-of-the-art facility here at what's called the Wexford building. And then you can see myself and my two partners down there at the bottom right, Panise Jasby, who many of you have seen, myself, and then Alex Mohr. Unfortunately, we don't get paid our living based on our smiles, so we don't end up doing a lot of pictures. But I want to explain to you real quick before we get into the really practical information about the microbiome. There's a 4-step process to working with us. The reason I put this up front is because I find that it's really helpful to have clarity around how to work with us if somebody decides to do that. I'm a very no pressure sales guy. So you're in what we consider on our end to be the training component of working with us. And so we train you, and the objective of that is just to give you enough information to make a purchase. We're not trying to be like, hey, here's how the microbiome works in every case that you'll ever come across in the future, right? That's unrealistic. You need experience. You need to understand how to utilize the test with patients. And so by the end of this call, I am going to try to tell you, again, it's not pressure at all. I only want you to buy if it's going to be helpful for your practice and for your patients and for your business. So if it doesn't meet those criteria, please do not purchase. But I am going to try to sell you at the end of this call, and that will bring you into the next step, which is hands-on training. And that's really where we get into the nitty-gritty of how this test works in your clinical practice and when you start to incorporate it. I find that we used to do hands-on kind of before the purchase. And what ended up happening is it would be really overwhelming. We're just giving you a ton of information and you're not actually grounding that in real life application. So what we did is hey, we just need to get you to the point to say is this going to be something that's helpful for my clinic? I'm not 100% sure, but I think it will. So I'm going to give it a try with a few. Then that's when we get into depth on okay, this is how you apply it. And then it sticks much better because people are getting hands-on experience with it. And then after that, you go into what's called our grow bucket. Again, these are how we as a business visualize our journey with you guys. And that grow bucket is continued education, continued support. And so, for example, the weekly webinars with Greg, if you guys have seen those, an onboarding program that we're currently building, all that kind of stuff would fit into the grow bracket of things. So without further ado, I'm actually going to give it over to Dr. Mohr here, and he's going to get into the microbiome and how it affects human health and how you can understand it.
Thanks, Micah, for the introduction. I'm sorry.
I muted them. It was somebody in the chat.
Okay.
Please continue.
Yeah, I appreciate the introduction, Micah. We are going to be going over the microbiome. Some of this information may be new to you. Some of it may be old. So we're just going to start kind of from square one in first looking at what is the microbiome, why we want to profile it, and then we're going to get into some specifics about what our test really offers. So if you're familiar with the systems biology paradigm, kind of looking at the host, we understand that we have the genome, we have the transcriptome, we have the proteome, we have the metabolome, which we have with our Aristotle test, looking at the blood metabolome specifically. And then we kind of have this black box, the microbiome. So the microbiome actually has its own metagenome, right? It has many different types of microorganisms that have their own genome. So when we say something like metagenomics or the metagenome, we're referring to this collective genetic biomass that resides in our gastrointestinal tract. So it really has its own scaffold of systems biology expression, each one of these omic layers from the genome down to the metabolome. We know when we look at expression over time, as outlined in that panel to your right, the metabolome is very sensitive to interventions, change over time as someone's physiology is changing. And we actually see this kind of mirrored with the microbiome. The microbiome can be very responsive, particularly when it's formalized in adulthood with things like, of course, antibiotics, but also dietary inputs to a large degree. So this really offers us a sensitive tool to look at what's going on in this environment. And we know, because it does reside in our gastrointestinal tract, that it's having a lot of implications into our own health. Now, the reason that I put a black box there is I think a lot of people have heard the term the microbiome, have kind of perhaps a more layman understanding of really what the implications of this system are. But as we'll go through these slides, we see that it's extremely complex. It is not easy to profile. And if you are profiling with a lot of current testing methodologies out on the market, a lot of the burden in terms of interpretation falls onto you. And that's really something that we wanted to address with our report and what we're actually doing with our analysis. So again, we're just going to go over some very high level information here in regards to the microbiome. So harbored inside the gastrointestinal tract at increasing concentrations down towards the colon resides a collection of microbes known as the gut microbiome. So really in the past, I would say 20 years, particularly as sequencing technology has really come into the fore, this has really become a hot topic. On, of course, the tailcoats of the Human Genome Project, we now have the Human Microbiome Project, which has had several iterations. And actually, we use some of that data in our own analysis. So the microbiome is very, very compelling for its deep connection to human health and its tremendous taxonomic complexity, but also, as Micah alluded to, its functional capability, which is really what we want to focus on. Gut microbiome testing is a specialized tool that provides unique insights into the composition and functionality into this ecosystem residing in our gastrointestinal tract. And unlike traditional lab panels, which often focus on specific biomarkers, nutrient levels, or organ-specific health domains, microbiome testing examines the interaction between gut microbes, the host, and then, of course, their collective impact on health, which we term the holobiont, meaning we're taking into consideration all microorganisms that are within or within the host, including the host. So microbiome testing is particularly valuable when we're trying to look at very specific root causes of different types of symptoms. Particularly, we know how prevalent gastrointestinal symptomology is, at least in Western populations. It's affecting the majority of people in some way, whether we're looking at the upper gastrointestinal tract or more commonly, the lower gastrointestinal tract. And we know a lot of this is actually mediated with the immune system. There is a tremendous amount of inflammatory signals passing from the gut microbiome into the host. This, of course, can manifest locally with things like IBS, but also systemically when we talk about circulating endotoxin affecting organs like the liver, and then even in the brain, which the gut-brain axis is very popular right now. So really, it's this very important system that can affect our health globally, all organ systems really, and it has tremendous. So just some brief A&P here. So really, when you talk about the gastrointestinal tract, while it is inside of us, if we actually go into the lumen, it's still considered an external environment. So there's a lot of protective mechanisms that the body has around this tract, and of course, a lot of internal organs. So we know that it's going to play a large role in our health. It's typically 20 to 30 feet. The majority of the population is going to experience some type of issue with their gut throughout their life. And of course, we know there are very important diseases that are specifically tied to profiling the microbiome. I'm not going to list them all, but you can look over that list. And I think most importantly, going back to what I was mentioning with being an important inflammatory mediator, approximately 70% of the body's immune system is located in or around the gastrointestinal tract, what we call GALT, or gastrointestinal associated lymphatic tissue. We know when we profile different sections of the gastrointestinal tract, we're going to see a lot of different conditions that are really going to dictate what type of microbes we see in these different sections. And the colon, or when we look at fecal material particularly, is very attractive because that's where most of the microorganisms are residing, because that environment is most conducive to their growth. Now, there are emerging technologies that we're exploring to sample other sections within the gastrointestinal tract. We're particularly interested in the small intestines. While microbial populations are at a much lower concentration here, they have a fundamental role in a very interesting area that we've actually done a lot of research in, and that is actually helping digest nutrients and, of course, absorbing nutrients, which, considering our food landscape nowadays, is an area of great interest. We also know that when we look at the microbiome, our minds generally go to bacteria. And while you look at a normal sample, at least from a fecal sample, you'll see about 95% of the microorganisms profiled are bacteria. There are many other constituents that form the microbiome, from viruses, fungi, archaea, and if someone is having some issues with, say, parasites, some multicellular organisms. So just to kind of put things in perspective, if we were to remove someone's gastrointestinal tract and basically dissect it and map it out, we're talking about 200 square meters of gut mucosal tissue. You also have to think how all the surface area that we have with the villi, of course, and the microvilli that we have on enterocytes. And this is effectively translates to a family house when we look at a blueprint, right? We're over 2,000 square feet. So I think this really is important for us to kind of, look at these very basic anatomical aspects of the gastrointestinal tract when we start thinking about interventions and what's actually going to move the needle on interventions. I know that probiotics are very popular. Probiotics have their time and place depending on the health condition of a person. But really, if you think about the biomass that's coming into the system with these, what should be targeted therapies, it's literally a drop in an ocean, as we can see with just how much surface area and how much biomass of resident microbes that we already have existing within that ecosystem. So if you are going to take any sort of treatment, really what makes sense is to perform a profiling to see what that environment is going to be conducive or receptive to in terms of intervention, regardless if it's dietary, pharmaceutical, or supplemental with something like a probiotic, a prebiotic, or other more emergent supplemental items out on the market like postbiotics, or even inoculated or dead zombie probiotics, as they're kind of more colloquially called. So there's a lot of different ways, from traditional testing to more advanced testing that we utilize here at Theriome to look at not only the gastrointestinal tract, but of course the microbiome. I'm sure many of you are familiar with these, particularly if you're at a certain age, right? We have things like fecal occult blood tests. We have fecal immunochemical tests, stool cultures, and now this emerging field of microbiomics. Of course, there's other ways to kind of get a proxy for gut health, even with breath tests to see, for instance, how much hydrogen you're producing, or if you are a methane producer. We can also look at some markers and make some inferences from what we see in circulation, whether we're doing a kind of standard enzyme link assays like lipid polysaccharide binding protein. That's a very popular one, and I actually think that still has utility. Or we can look at something like calprotectin in the fecal sample itself. There's also food allergy and sensitivity tests that are available. And of course, metabolomics, which you can sample the blood and make some inferences on microbial derived metabolites. And of course, you can also profile the fecal metabolome, which we feel is going to be very, very important in really crossing the bridge with integrating what's going on in the microbiome and what's going on in the host. So I'm just going to briefly cover some technologies that fall under the domain of microbiomics. Really, the workhorse of profiling the microbiome right now in terms of sequencing is all predicated on 16S ribosomal RNA amplicon sequencing. So for this, you're using a series of polymerase chain reactions, amplifying this specific, basically, gene, it's RNA. What we're looking for is actually regions that are hypervariable in the specific gene, not those that are conserved. So this is what's going to help us identify different microorganisms. So again, we're making a lot of inferences here. There's different hypervariable regions you can profile. Most normally, when we're looking at human fecal samples, we're looking at the V4 region. However, we now have new technologies like the PacBio system, which is able to look at all 9 V1 through V9, so we can perform a full-length survey of these regions, and we can get hit or miss down to the species level. So this is a feasible option. I know that many companies are using this. However, when you're using this technology, as I mentioned, you're making a lot of inferences. You're working with databases that are actually performing these classifications. And in most cases, they're not using the long read sequencing. They're looking at a specific region like V4. And really, with confidence, you can only get down to the genus level. Now, in terms of functionality, the issue is because we are using this particular gene for annotation, we are not able to confidently, with great confidence, I mean, above really 80%, confidence, annotate the functions of the microbe. I know there are tools available like PyCrust, Taxa for Fun, that can make some inferences, but their confidence is generally 70, 80%, and really they're limited by making those inferences at the genus level. So it's a good starting point for large-scale population studies. It's good to start with this because then you can start doing some hypothesis forming, but in terms of actually being very granular and very specific, this is kind of an antiquated method for individual testing. Really, the most accurate testing currently available would be whole genome sequencing. We generally call this a shotgun approach because effectively what we're doing is we're taking all the DNA from a viable sample. So that's another issue that we can get into a little bit later in terms of collecting the most robust, accurate snapshot of the sample, really with our own pilot testing internally and the clinical research that we've been involved in. We've actually found that, unfortunately, it's not just as easy as taking a very small swab. It's not just as easy as taking a fecal wipe. We've run into major issues with not getting good biomass in our sample. So really, we might be missing a lot of things. And we want to get a very holistic sample. So we actually utilize a special patented collection method, the OmniGut, offered by DNA Genotech. Unfortunately, it does require a bit more sample volume, about a pea size taken from the internal of the stool expression. And that is going to give us enough biomass to perform our whole metagenome sequencing, where we're breaking down the whole genome into these short random fragments. We then sequence them, all these little fragments. It's a very computationally intense process. And then we assemble them, we reassemble these short reads into long contigs. And this reassembly project effectively rebuilding the whole genome from these short fragments. And then we're building out what is actually there taxonomically. And then because we do have the DNA, what they're doing functionally. So just a brief side-by-side, 16S sequencing is inherently less expensive. And this is why a lot of companies are utilizing this. There are shotgun methods available where you're doing shallow sequencing. You might be looking at anywhere from 1 to 3 million base pairs. That's better than 16S. However, it's not going to be as revealing as deeper sequencing like what we use with our test, where we're looking at a minimum of 10 million base pairs. For 16S, the taxonomic resolution generally is going to be limited to the genus level, unless you are utilizing PacBio's long read system, which with some confidence you can get down to the species level. With deeper sequencing, looking at using the shotgun approach, we are able to get down to the species and even the strain level, which is really important when we start tying in functionality and understanding what specific microbes, but really what they're doing, right? We are also able to detect things like plasmids, phages, viruses. Now, in terms of tool building, of course, internally, we had to build out a tremendous amount of bioinformatic infrastructure to handle this, because while there are plenty of tools available for 16S, it's a bit more variable for deep sequencing because there's simply not as many people using this methodology, and it is a lot more expensive. The other issue with 16S is we can have contamination, whereas when we're profiling the entire metagenome, we're actually able to bench that. Actually, our own tools using Bowtie, where we index that against the human genome and actually remove any host-associated DNA so we don't get that contamination and make spurious inferences based on particularly functionality. So what our test offers, now I think there are plenty of really good tests out there, particularly depending on how someone is utilizing their test. There's always going to be different use cases. If someone's looking at a very specific of inflammatory markers, or say they're only interested in looking at a proxy of gut permeability like calprotectin, that's totally fine. But really, if you want to get a very detailed view, we feel like our test is bringing something pretty unique to the market. So first of all, because of the type of sequencing technology that we are using, we are able to get to the strain level. So looking at the tree of life, phylogeny, we go down from kingdom, if we're using something like 16S, to genus, if we're getting a bit more resolution, species, and then obviously strain. So this is kind of the terminus of the tree of life, and really important. We really want to understand the full dynamics and the full ecosystem of the microbes. This is more a comprehensive profiling. Also, because we're able to more definitively capture functionality, we can perform robust pathway analyses to see what is functioning well and what might be perturbated, and really what we need to address, rather than relying on drawing associations from the literature with a specific microbe, say like C. diff or something. We actually want to see how it's operating in the ecosystem. And to do that, we form 15 key analytical domains using our in-house bioinformatic pipeline that I mentioned, which does utilize artificial intelligence. Simply, when you are doing these analyses, this is not as simple as a clinical blood work where you have a set number of markers, you might have clinical experience. The number of features that we're dealing with, the human brain simply cannot at least on a rapid time scale when we're talking about patient interactions, what we need, fully digest that well and make the appropriate, inform appropriate interventions and appropriate treatment for their patient. So really a nice option about microbiome tests in general is this does not really require a lab visit. This can be done in the comfort of their own home. We understand that, and I have been, jabbed at endlessly for my involvement with the microbiome, just simply based on the type of sample that I have to deal with on a constant basis. But this whole ordeal, it can be a little invasive for people. So doing it in the comfort of their own home is very, very nice. As I mentioned, we utilize the OmniGut test from DNA Genotek. So this has unique stabilizing chemistry within the actual collection device. So after the person basically has their expression, collects their pea-sized sample and puts it in the tube. At room temperature, that sample is viable for approximately 2 months. Obviously, we would like them to get their sample in as quickly as possible, but because of the proprietary chemistry that DNA Genotek is utilizing in this particular device, we are very confident in our own internal pilot testing, and their published data shows a very viable sample for us to actually survey in our lab. So I just put very simply, we collect the sample upon the person sending their sample in. You have about a 2 or 3 week turnaround time right now. And really the secret sauce of our report and how we're different from a lot of other companies out on the market is we have different layers of translation, obviously more high level. And as we go throughout the report, it gets a lot more granular. We also have support with myself and our other health translators that help you understand how to interpret the report and then also translate these findings to your patient. We are profiling at the strain level, the functional level. We're developing personalized protocols. You don't have to, obviously you want to bring in your own clinical knowledge, your own intuition, but you're not having to perform all these univariate analyses based on which microbes you're seeing, what functions you're seeing. We actually do a lot of the heavy lifting, and as I mentioned, this is across 15 different domains. So looking at some of the internals of the report, similar to the Aristotle, we perform this kind of global analysis. We're looking at an overall health score, incorporating all of these domains, which I'll get into. So this is kind of your dashboard to see where you are kind of grossly. Looking at some high-level summaries that are at the first part of the report, we do have a conceptual summary, which I'm a very visual person. I try to incorporate a lot of visuals, and that is something that we are continually improving in the report, and of course, working with our physicians that we have that have already purchased this test and providing their own input. But we're kind of doing broad strokes and what are the problematic areas that someone, not only based on their survey data that's informing a lot of the analyses, but what's going on internally in terms of the heavy hitters and what we can address to really move the needle in this person's health. We also perform a global analysis, kind of looking at what are the main constituents of this person's microbiome. In this particular instance, we obviously see their microbiome is dominated by bacteria, to a lesser extent viruses, and it's at such a small degree we don't see some of the protists and archaea that are present in this sample, and we do break out into greater detail, but this is just giving us kind of a gross look at the microbiome. Very uniquely to our test, we are actually utilizing a recently published data set from over 8,000 metagenomes or 8,000 microbiomes that were profiled using whole genome sequencing. So we're actually able to benchmark your overall wellness index, as they term it, but really you can more liken this to a dysbiosis score. So this is a very broad metric that you can kind of use as a proxy to see shifts in someone's overall microbiome based on the intervention that you're giving over time. So depending on the recommendations that we're going to give for retesting, we would want and we would expect to see this score increase where someone has a more health-associated microbiome. And this particular analysis, which we provide scientist notes for, so we interpret why you might have a low score, why you might have a high score, looking at the specific microbes and their functions that are involved, really driving this. Beyond this, we also have some pretty standard ecological metrics, obviously looking at the keystone features or the core microbiome of someone's overall sample, seeing what really are the key drivers of that environment, and really what might be nodes of what we want to address in terms of our proposed therapies, because those are going to be really the more resilient microbes, whether they're healthy or even disease associated. We also provide benchmarked against the Human Microbiome Project, or the Human Microbiome Project, pardon me, where they fall in relation to what we term alpha diversity, or really how feature rich that environment is. Now, in the past, it was seen that high levels of alpha diversity or greater scores of alpha diversity was really a proxy for health. And what we're coming to understand is it's really context dependent. You can actually have someone that is severely constipated, have very high alpha diversity, but they're not really in a health state. So you really have to pull in all these different data points and form a bigger picture and perform a more cohesive and encompassing analysis, which is something that we do. We also have our dissimilarity score, or beta diversity, really how unique your microbiome is in regards to the population. Now, this is something that can be important, particularly if someone has some different environmental exposures, depending on where they are geographically, who they're around, all these important factors that we capture in all the important survey data that we have informing a lot of these analyses. Also unique to our test is a resilience score. So resilience is important when we talk about any biological system. And generally, I know that this has gotten a lot of traction in psychology, talking about like mental resilience, for instance, but this can really be applied to any biological model. And what we know in terms of someone that might be in a dysbiotic state or a disease state, they can actually have a very resilient microbiome. So it's not necessarily a good thing. And really what we want to do with that person is identify the anchors, the attractors that are making that microbiome resilient if it is dysbiotic, and kind of help release those anchors so we can transition the microbiome into a more healthy state. So really, in many ways, we're talking about a bottleneck event. We actually have to address certain populations of microbes, try to reduce those, and then increase beneficial, again, context dependent. There's not universally beneficial bacteria or beneficial microbes. It really depends on what that person's microbiome is going to be conducive to, but put them into a more health-associated configuration. We have subdomains within this informing the score that we are really addressing in terms of the interventions that we recommend through a little bit later. We also look at some other really important kingdoms in the microbiome. I know everyone gets fixated on the bacteria, but of course, they're dominant, but they're just one side of the story. So we also profile the archaeome, and we perform neural network analyses where we're looking at the functional nodes that they're associated with. So this really helps us, as I was going back with resilience, kind of identify those anchors that if we were going to address someone in dysbiosis or someone in a disease state, what those particular features are that we need to address. The microbiome, I'm sure this is something that everyone has heard of, again, on the call, another kingdom that's very, very important to profile. Beyond the neural network analyses that we're performing here, we're also including mycotoxin bioremediation capacity scoring. So mycotoxins are toxic secondary metabolites produced by fungi that frequently actually contaminate food and feed that we're giving a lot of our animals, impacting estimated about 60 to 80% of the global agricultural food system. So this is actually a really emerging area, and I felt very important to include in our test, particularly as we understand, you really are what you eat, and we really want to get a handle on how your microbiome is dealing with these. And in many cases, while it would be nice if we lived in a perfect world and we didn't have these exposures, some of these are kind of omnipresent in our environment right now. And really, we need to get a firmer handle on how to address dealing with them, whether that be, looking at our liver health, but also how we can leverage the microbiome to handle some of this, particularly in terms of the specific functions that we're looking at are informing this panel. The virome. Of course, viruses are really important custodians for the microbiome, really modifying not only overall broad ecological metrics, but also bacteria and other microorganisms themselves. So actually, the virome can be leveraged for good or for worse to really affect all kingdoms of the microbiome. So this is another layer, a kingdom layer that we're including in our test. More traditionally, we do have a probiotic panel. There's elements of our innovative approach but then also kind of checking the boxes on what some other panels are offering. I think this is really important, particularly when physicians or practitioners are looking to onboard our tests, but still want to have some of the features of other tests that they had used in the past, kind of as a bridge. So we do have a pretty encompassing probiotic panel that we are expanding. So this is important when we're actually going to inform probiotic therapy, if that's warranted, and then also to see over time if we're actually seeing these probiotics effectively work in the entire ecosystem. A really important confirmatory rather than just taking random probiotics. In a similar vein, we also have a pathogen and parasite panel. So this section identifies and measures different pathogens, as well as parasites that are known to influence gut health. It's important to note that while these organisms may not be present in the gut, for everybody, and I know how popular these are right now, it's kind of a nice piece of information to have just to see if there's anything underlying that more broad microbiome tests are kind of failing at. I know there's several products on the market that currently profile this as well, albeit using different methodologies. We also have a antimicrobial resistance panel. So this panel comprises a total of 814 amplicons to assess the presence of 478 antimicrobial resistant genes across 25 different antibiotic classes. So we all know that, particularly if you're living in the West, it's likely that you've had pretty broad exposure to antibiotics. So this is important to profile not only in future antibiotic use, but also in looking at alternative therapies, if that is warranted. I know there are actually some really well-validated, more traditional remedies that can do a lot of these things. And if someone is having issues with certain classes of antibiotics, find some alternate routes. Of course, if we're talking about the gastrointestinal tract, we know how important the microbiome is to digestion, as well as nutrient generation. So these we feel like are fundamental to including in a microbiome test because we do have the capability of fully profiling functionality. So here we're looking at important things like short-chain fatty acid production, which is very important, particularly when we talk about, say, butyrate being one of the principal sources of fuel for enterocytes, or the cells that actually line our gastrointestinal tract, as well as other really important signaling agents like acetate, which can actually tie into liver health and adiposity, as well as the myriad of bile acids and the different types of microbes that can effectively deconjugate some of the primary bile acids that are then coming into lower regions of the gastrointestinal tract, and in some ways can wreak havoc or be leveraged for health, particularly when we talk about communication in the gut wall with bile acid receptors that affect glucose and lipid metabolism, like the FXR receptors, TGR5, et cetera. So those are all included here, as well as looking at some key vitamins, for instance, like B vitamin complex, as well as K, which we know the microbiome contributes a substantial amount in terms of actually creating that particular class of vitamins. We also have a gut barrier integrity panel, which is informed by some subdomains. So having a disease-associated microbiome is one thing, but really we want to know how it is potentially affecting the host, the local level. And really the main gatekeeper, of course, is our gut barrier. So we really need to get a handle on how that is performing, and if we need to address that, whether changing the microbiome or also doing some therapies for the host to improve the integrity of those specific cells, the human cells in the gut barrier, as well as, and relatedly, an inflammatory panel, because we know how the microbiome is really one of the main culprits in terms of spurring inflammation, whether it be acutely or even contributing to meta-inflammation that so many people have issues with, and is effectively tied to all chronic disease in some form. And after this, we do have a more traditional component of the test looking at keystone microbes. Again, in many ways, what you would see in other tests on the market. This is really a feature of incorporating the new and the old to have components of the test that might be more familiar to you, what the implications of these different microbes are, what we're seeing in terms of the result, and then where you're following in reference to a large pool of metagenomes comes from other microbiome samples. So beyond the analytic domains that we have, really one of the main features that we have in our test is our digital twinning, where we're effectively able to create a digital replica of the patient based on their own microbiome data. So we are constantly mining the literature to pull effective interventions. This is really a full-time job in many ways because the literature is fast moving and there are a lot of really new therapies coming into use, a lot of different supplements, dietary factors, lifestyle, sleep as well is very important. And even environmental exposures, whether it's based on geography, water source, even if you're living around animals, for instance, what your significant other is doing, all those are really important and captured by our continuous crawl through the literature to really inform our protocol. So we perform based on the digital representation of your data, 1,000 different simulations, and we're able to get very specific with the type of protocols that we design in our report. So these are recommendations. Obviously, in working with me in training or other people on our team, we can better teach you how to leverage this tool and to an extent customize it for your particular use case. However, we're not replacing you, obviously. You still need your own interpretation of the data and the specific patient, but this takes a lot of the burden off of you looking at potentially thousands of different features and trying to make sense of it. Really, this is pattern recognition, something that the human brain just can't efficiently do anyway, and make your job a bit more easier. So these personalized protocols are domain specific. We also recommend, based on our analysis, when to retest again to see a shift in the scoring that we have by domain for a person. And this is scored in terms of impact, really what's going to move the needle. So rather than just give you, a whole grab bag of potential therapies or treatment options, really show you what you want to focus on. And if you so choose to go down to some of the lower tiered recommendations, whether they have a lower impact based on them not moving the health needle, so to speak, as some of the more high impact factor therapies, or even if they're not as, say, well validated in the literature, they are there and you can use them. So I'm not going to go over all of the potential use cases for our test. I think it's fairly apparent, and I mean everyone's going to be different in terms of what their practice is or what their health goal is. But this obviously can be utilized for a wide array of really gastrointestinal, and then even looking at some things that might be gastrointestinal organ but affecting other areas of the body. Really, it's open season, and we're really excited actually next year to start performing a true multi-omic analyses where we're integrating different reports together. So data from what's going on in circulation with our Aristotle test, looking at the metabolism of the human body, and then also what's going on in the microbiome to really get very granular in terms of really translating this ecosystem into broad human health and getting actually organ specific. So with that, I will hand it over back to Micah, and I'm happy to take any, if you want to contact me, happy to continue the conversation.
Thank you, Alex. I really appreciate that. So I'm going to wrap this up. There was one quick question in the chat here, Alex. Do clients need to quit taking supplements prior to testing?
No, they don't. The main thing, though, the survey that we have is, I will tell you, it's fairly detailed. We really urge, if you're going to take this test, I know it's going to take a little bit it's not that intensive, but I mean, it is going to take some time, but really to make our test the most robust it can be, they need to be fully transparent with everything that we're doing. And we ask for medications, we ask for their dietary data, we ask for their current supplement regimen. Just as long as they're being detailed and fully transparent with that, that is enough.
Fantastic. Yeah. So you guys might have seen in the chat, I put the link to the sample report. So Alex covered a lot today. It was pretty high level on what is included in this report. But from this, there were a few things that stick out. Number one is that he's using metagenomic sequencing over 16. I don't know if you guys caught that, but with 16S, what he was saying is like, you really only confidence down to the genus level. So what does that mean? That means you're really not getting visibility into what is actually there, what is actually present. And that's kind of the standard that's currently utilized. So we're upping that standard with a deeper resolution, getting down to strain level, also including things like the microbiome, which if you didn't catch, that's fungi, and what they're putting off. And so some really good stuff there. Take a look at that link. But right now, what I'm going to do is I'm going to just go through what we're offering. This is the early access launch. So what does that mean? It means that today we're going to be doing 6 of these webinars. So if you want, you can get this offer 6 times, but you have to sit through for an hour before you get it. But over these webinars that we're going to be doing over the next week is that we're only offering this right now. We're not going to do a full launch of the microbiome test until sometime next year, kind of looking in the February timeline, but you're looking at another 4 or 5, potentially 6 months before you'll get access to that again. So we're onboarding practitioners right now in our early access launch. Once they get into that, they can buy as many as they want, but you can't access it again until we get to our full launch where it'll be available to everybody. So my slide's not working. There's my lock. All right. So early access opportunity, I'm going to tell you what that's going to entail. So I approach this from the standpoint because I was trying to draw parallels to what currently exists in the marketplace. And I really wasn't able to come up with a one-to-one comparison. It's kind of apples and oranges mostly. You saw some of the things that he's bringing up, like the microbiome, the archaeome, the parasites, all these different elements that Alex is bringing together. So what I did was I went ahead and said, okay, what would somebody need to do if they went into the market to get the same thing today? If they tried to replicate this, and I even discontinued the idea of digital twinning. I discontinued the idea of the microbiome. I discontinued the idea of the archaeome, but like roughly like there's some generalization ballpark that we get to. So does anybody know how much you would have to go through to get a similar amount of data? Well, you would have to get the Viome test, the GI map test, Viome at 249, GI map at 359, Genova Diagnostics parasitology profile, Genova Diagnostics GI effects profile, and then you would have to get a BioFire gastrointestinal pathogen panel on top of that. So what is the total? And that's just to get to the data and not even all of it. The total that you would spend to get that is over $2,000 for less information delivered in a more complicated way. So that's what I was saying. I was really blown away with what Alex has been developing over this time, is because he's really solved a big market issue in consolidating a lot of information in a very easy to understand way, coupled with protocols that tell you exactly what to do about it. And again, that, if you've been through some of our webinars, when it's coupled with a practitioner, is going to be much more effective. But that protocol, so let's get into how much time, again, if you're going into the market and you're trying to get a similar thing that doesn't include a lot, but roughly similar, how much time would it take you per patient to get a similar result? Well, you would have to get those 5 tests. You'd have to do 5 different sample collections. You'd have to review 5 different reports, researching the connections between the results and interpreting for actionable insights. I generously put the amount of time spent to do that would be 5 to 8 hours. Most of that time would be spent researching the reports and translating what is the significance of this particular taxonomy, this particular microbe, this particular thing. So there would be a lot of time spent in just understanding what Alex has just presented right in the report. Like, hey, here's your resilience. And he didn't even get into the subdomains. You could go through his report and ask him many questions deep on any particular domain, because there's also the subdomains underneath it that tell you like, hey, your resilience is 80% of its capacity because your nutrient generation is such, your et cetera, et cetera. So the next step then you would have to do after going through those reports is protocol curation and development. This would be cross-referencing data, researching the microbiome for systematic connections, adjusting for dosage timing, sequencing based on literature and clinical experience, and accounting for unknown interactions between interventions, generously put 1 to 3 hours. Patient communication, you have 5 different reports. There's a lot of things that they're just very clinical. They're just saying, here's the species or the genus and the population of that thing. So you need to spend some time communicating that to the patient, depending on your style, I put 30 minutes to 2 hours, considering that it's 5 different reports that you'd have to go through. Maybe you're a bullet point type of person and you blow through it in 10 minutes. I don't know. So I'm estimating that time saved per patient is approximately 6.5 to 13 hours. So for 5 patients, that would be 32.5 to 65 hours of time saved to get roughly a similar thing, not including a lot. So when you take us up on this offer, this comes with state-of-the-art training by Dr. Mohr. He is an expert in microbiology. As you can tell, he has a very deep understanding of this, and we fully plan to leverage that for your benefit. So unparalleled expertise in microbiome science. So like I said, expert. So what we're providing is, if you take us up and become a practitioner with Theriome today in the microbiome, is you get state-of-the-art training directly from Alex. So you're going to get a one-on-one with Alex, going over the report, going over patient results so that you can understand it in more depth. And then thereafter, we have Greg Nacarelli, who is a PhD MD, also with a very good understanding. And if you can use it as much as you want. So you'll get Alex, but you'll also get access to some of the other scientists on the team to whatever degree you want. I put, Alex, it's hard to put a dollar value on him, but I had to for the point that I'm trying to make here. I estimated around 500 per hour. This is coming from my business experience, specifically in this space, what I would expect to hire somebody in of Alex's caliber to consult with us and develop something like this. So you gain an additional $3,000 of value just in the training and the consultation for no additional cost beyond the test kits. So let's get into the cost. What is this going to cost you? Well, when we do the full launch, it's going to be $800 per test. Today, it's $600. So you're saving $1,000 when you buy 5. The first bonus, you get 5 tests in one. So like I said, it was over $2,000 to get all 5 of those tests and just to get something similar to what the Élie microbiome test that Alex created has. So that'd be $10,000. Then time saved, 32 to 65. I took 40 hours, just kind of put it on the low side of average and said $100 an hour. So you save $4,000 in time. And as you know, if you're a business owner or a clinician, it's very, very valuable. And then bonus number 3, you get trained by a world-class expert in microbiology. I put $3,000. If you guys have been in master classes or through courses or different things that can be way more, I put 3,000 because it seemed pretty fair. So total value to get something similar would be over $18,000. What are we offering it for today? You do have to buy 5 of them. Any guesses? $3,000. I think it's a steal. This retails for $5,500. So today, if you get 5 of them, and you have to buy 5, you can't buy 1. For the early access, the reason we're doing that is because we want to have a consolidated amount of practitioners that we're working with that are working with multiple patients on the microbiome, because they're also going to be working with Alex. And Alex has limited bandwidth, limited amount of time that he can do, but he's also going to be deriving from your clinical experience and your understanding of the test to make further improvements and iterations. So the feedback you give can actually make it into the test in the future as well. So price for today is $3,000 to get 5 of them. And the way you do this, again, this is only through this webinar. You won't get this again until we do the full launch. But you have to put your email in the chat right now if you want to get an invoice. And so what I'll do then is I'll send you an invoice. Once you pay it, you'll get your 5 kits, and then we're off to the races. But you do have to put your email into the chat today. And that is how you get started with the Élie microbiome test. So I know we're running up on the hour here. Again, if you are interested in getting started, please just put your email into the chat here. Again, this is only available through one of these webinars, and you'll get access to Dr. Mohr.